First week of human development. After fertilization, a zygote is formed, which contains 46 chromosomes. The newly formed zygote develop repeated mitotic divisions, a process known as cleavage. The first division occurs after 30 hours, and it results in the formation of two cells. Each one is known as blastomere. Cleavage occurs as the zygote passes along the fallopian tube. The second division results in the formation of four cells, and repeated divisions results in smaller cells. As you see, blastomeres in the 4-cell stage are smaller than blastomeres in the 2-cell stage. At 4-cell stage, the zygote activates its genetic material, a process known as zygote genetic activation. Initially, the zygote depends on the maternal transcripts and the proteins. However, these materials are eventually depleted, and the zygotic genes are turned on. Now the embryo starts producing its own proteins and RNA molecules. Zygotic genome activation is a critical event in development, and any defect in this process can lead to miscarriage, birth defects, or other health problems. The next step is compaction and morula formation. After the 9-cell stage, blastomeres become tightly aligned against each other, a process known as compaction. As compaction progresses, blastomeres flatten against each other and establish junctions between them. This results in a loss of distinguishable boundaries between blastomeres. During compaction, cells differentiate into two types of cells. The outer cells form the trofoectoderm, while the inner cells with tight junctions will form inner cell mass. After compaction, the developing embryo is now called morula, which is typically seen around 4 days after fertilization. Cavitation is the next step. The trofoectoderm come back together and form junctions, capable of transporting ions and water inside the morula. Shortly after the morula enters the uterine cavity, about 4 days after fertilization, a fluid-filled space known as blastocystic cavity emerges inside the morula, and now its name changed into a blastocyst. This process is known as cavitation, and it takes place between 16 and 32 cell stage. Now the blastocyst is formed of an outer trophoblast, an inner cell mass, a blastocystic cavity, and is surrounded by zona pellucida. The early blastocyst on day 4 or 5 initially does not increase in size, and the blastocystic cavity represents less than 30% of its volume. Over the next 1 or 2 days, day 5 or 6, more fluid accumulates inside the blastocystic cavity, and finally it forms about 70% of the embryo's volume. Blastocyst expansion is a dynamic process characterized by oscillating contraction, blastocele collapse, and recovery, which occurs at intervals of 2 to 4 hours. As the blastocyst expands, the zona pellucida becomes thinner, and finally, hatching will happen. Hatching means that the blastocyst will free itself from the zona pellucida. As you see in this picture, this is the zona pellucida, and the blastocyst started to escape from inside the zona pellucida. Hatching is completed within a few hours, resulting in the blastocyst being separated from the empty zona pellucida. And now the blastocyst becomes capable of implantation, which will be discussed in the next presentation. Thank you.